Imagine a scenario that sounds, well, it sounds a bit like something straight out of a medical thriller.
Oh, I like a good thriller.
Right. So a patient is enrolled in a clinical trial for a very powerful new medication. And on Monday, they're administered a fixed dose of this drug at exactly 8:00 a.m.
Okay.
They tolerate it perfectly. It does exactly what it's supposed to do.
Mm.
But then on Tuesday, another patient, exact same age, same weight, identical clinical profile, receives the exact same dose of the exact same drug. But they get it at 8:00 p.m.
12 hours later.
Exactly. And for that second patient, the dose is lethal. The toxicity has suddenly spiked by a factor of 10.
It sounds completely impossible if you view the human body as a static machine, doesn't it?
Yeah, you'd think it would be exactly the same.
Right, because if the kidneys filter at a constant rate, or if liver enzymes metabolize things at a constant speed, a drug should behave the exact same way, regardless of what the clock on the wall says.
But the body isn't a static machine, and that is the illusion we're sort of shattering today. So welcome to you, our listener, to this deep dive. Today, we're exploring the temporal architecture of the human brain.
A fascinating topic.
It really is. We are acting as your companion to Chapter 1.12 of Kaplan and Sadok's comprehensive textbook of psychiatry. And before we go any further, we need to establish a foundational anchor, really, for how we approach this material.
And that anchor is that psychiatry is fundamentally the study of how the brain, the mind, and experience interact. And crucially, how that process can go wrong.
Right. And that interaction is dynamic. It's constantly moving.
Yes, exactly. For decades, medicine has largely focused on the spatial aspects of biology. Where is the lesion? What is the specific receptor? Which neurotransmitter is depleted?
Very geographical.
Spot on, geographical. But to truly understand psychiatric vulnerability, we have to look at the temporal dimension. We really have to start asking when.
Okay, so to visualize this, I want you, the listener, to picture the brain not as a computer with, wires and hard drives, but picture it as a sprawling, highly disciplined symphony orchestra.
I love this analogy.
It works so well. So your organs, your cellular processes, your neurotransmitters, they are the individual musicians. You've got the violins of the cardiovascular system, maybe the heavy brass of the endocrine system, the delicate woodwinds of cognitive processing.
Right.
Now, deep inside the brain, there is a tiny cluster of cells called the suprachiasmatic nucleus, the SCN. That is your master conductor. And environmental light, well, that is the conductor's baton.
It's an incredibly elegant framework because, think about it, consider a liver cell. A hepatocyte already knows how to process toxins. It doesn't need the brain to tell it how to do its job.
Just like a violinist knows how to play the violin.
Exactly. The adrenal glands know how to synthesize cortisol. The musicians know how to play their instruments. But without the conductor, well, the violins start playing before the cellos. The timpani crashes right in the middle of a silent pause.
The individual parts work, but the timing is completely lost.
Total chaos. Your metabolism ramps up when you're trying to sleep, or alert signals fire when you are absolutely exhausted.
And that resulting cacophony, that profound loss of temporal harmony, is what we often recognize clinically as psychiatric and sleep disorders.
Which is our objective today. We're going to trace the trajectory of this phenomenon, from the microscopic molecular gears inside that master conductor all the way out to the clinical manifestations in human behavior.
We're shifting the paradigm from the anatomy of space to the biology of time. So let's start at the very foundation of chronobiology. Just breaking down the word itself, circadian gives us a clue, right?
Yes, it comes from the Latin, circa meaning about, and dies meaning day. So about a day.
But, I always assumed that meant precisely 24 hours. The Earth spins, the sun comes up, 24 hours later it happens again. We evolved on this rock, so our biology should match it perfectly.
You would certainly expect an evolutionary mechanism that's conserved across nearly all living organisms. From cyanobacteria to fruit flies to humans, to be perfectly precise.
Yeah, evolution is usually pretty ruthless about efficiency.
It is. But the mammalian internal clock, which is generated by the SCN, has a fascinating quirk. It does not run on a perfect 24-hour schedule. In humans, the endogenous rhythm actually runs at approximately 24.18 hours.
Wait, I have to stop you there because that makes absolutely no sense from a survival standpoint.
It seems counterintuitive, doesn't it?
Totally. If we are off by almost a fifth of an hour every single day, if my watch loses 11 minutes a day, eventually I'm eating lunch at midnight, wouldn't early humans have just drifted completely out of sync with the sun?
If left entirely to their own devices, yes, absolutely. And we know this definitively because of isolation experiments.
Oh, like the cave experiment.
Exactly. In the mid-20th century, researchers like Michel Siffre spent months living deep in subterranean caves, completely isolated from sunlight, from clocks, or even variations in temperature.
Just total sensory deprivation regarding time.
Right. And in the absence of any environmental cues, the human biological clock free runs. It just does its own thing. So Siffre drifted slightly later, every single day, eventually living completely out of phase with the actual day on the surface.
So his internal 24-point-100-hour clock just kept ticking unapologetically.
Precisely. But, in the real world, we don't live in pitch-black caves.
Thankfully.
We don't drift. And this highlights a crucial evolutionary design principle. The clock is inherently slightly flawed in its native period, but it is highly malleable. It is designed to be corrected.
So it requires constant recalibration to the 24-hour environmental reality.
Spot on. And it achieves this through what we call Zeitgebers.
Zeitgebers, that's a German term, right? Literally translating to time givers.
Time givers, yes.
Yes.
These are the environmental cues that effectively rewind or fast-forward the clock just a little bit every day to maintain the proper phase relationship with the Earth's rotation.
And light is the big one here.
The primary, overwhelmingly dominant Zeitgeber is light. But the system is robust. It monitors non-photic Zeitgebers as well.
Like what?
Well, the timing of your meals, your social interactions, fluctuations in ambient temperature, and when you engage in physical exercise.
So the brain is just aggregating all of this data to anchor that wobbly 24-point-8-hour rhythm strictly to the 24-hour day.
Exactly.
It's sort of like having a vintage mechanical watch that runs slightly slow. It's not perfect, so every morning when you wake up, you glance at the sun and just manually adjust the dial back a few minutes.
That's a brilliant way to put it.
Yeah.
And this endogenous variation, the fact that the watch runs at its own idiosyncratic pace, that explains why we have larks and owls, doesn't it?
Oh, right, because the 24-point-8-hour figure is just a population average.
Exactly. There are genetically determined variations in that endogenous period length from person to person. So imagine someone whose natural internal clock runs a bit shorter, say, closer to 23.8 hours.
Okay, so they're naturally faster than the Earth.
Yes. They're constantly phase advancing relative to the environment. Their biology is always pulling them to start the day earlier. That individual is your classic morning person, a lark.
And the owl is the exact opposite. Their internal clock might run, at 24.4 hours. So their biology is constantly dragging them later into the night.
Precisely. It is not a moral failing or laziness, which is how society frequently pathologizes it.
Especially in teenagers, right?
Oh, absolutely. Teenagers' clocks naturally phase delay during adolescence. It's a structural reality of their biological oscillator.
A profound biological reality. Okay, but to truly understand how this oscillator works, we need to look at the hardware. We need to examine the anatomy of the master pacemaker itself.
The suprachiasmatic nucleus.
Let's locate it for our listeners. If I'm navigating through the brain, where exactly am I looking?
You are heading deep down into the anterior hypothalamus. The SEN rests just dorsal to meaning immediately above the optic chiasm.
And the optic chiasm, of course, is where the optic nerves cross over one another.
Right. So anatomically, it is perfectly positioned to intercept visual information coming straight from the eyes. Structurally, it consists of two paired teardrop-shaped nuclei.
Okay. And the cells inside, are they just your typical neurons?
They're quite distinct, actually. The SCN contains some of the smallest neurons in the entire human central nervous system. They are incredibly densely packed, and they possess short, relatively unbranched dendrites.
So they aren't reaching out across vast distances with long, spindly arms. They are a very tight-knit community, talking constantly amongst themselves.
A highly synchronized community. And we can functionally subdivide this nucleus into a core and a shell.
Okay. What's the difference?
The core is defined by the presence of calbinin-positive neurons. Calbinin is a calcium-binding protein, and its presence essentially helps us map the functional geography of the nucleus.
Right.
The core receives the bulk of the environmental input, and it then relays that information to the surrounding shell, which generates the rhythmic output.
So let's trace that environmental input, because this is the part I find fascinating. How does this tiny, dense cluster of cells buried in the dark center of the skull actually see the light to know what time it is?
It relies on a dedicated, highly specialized pathway called the retinohypothalamic tract, or the RHT.
The RHT.
Yes. As the optic nerves carry visual information from the eyes toward the visual cortex at the back of the brain, a small bundle of fibers just branches off at the optic chiasm and projects directly upwards into the SCN.
But here's the amazing part. This tract isn't using the normal rods and cones that give us conscious vision, is it?
No, it's not.
So you could theoretically be entirely blind, completely unable to see shapes or colors, but still have a perfectly functioning circadian clock.
That is a critical distinction, yes. The RHT originates from a distinct, very small subset of ganglion cells in the retina. We call these intrinsically photosensitive retinal ganglion cells, or iPRGCs.
Bit of a mouthful.
It is. But they contain a unique photopigment called melanopsin. And melanopsin doesn't care about the shape of an apple or the text on a page. It is highly, uniquely sensitive to a specific wavelength of light.
Blue light.
Exactly. Blue light peaking roughly around 480 nanometer.
And there it is. I mean, this is the reason our modern world is just a chronological nightmare.
It really is.
For millions of years, the only source of bright blue light was a cloudless daytime sky. So melanopsin evolved as this highly reliable daytime detector.
Yes.
But now what do? We stare at smartphones, tablets, and LED televisions at 11:30 p.m. We are literally blasting our retinas with the exact wavelength of light that tells the iPRGCs, hey, the sun is high at noon.
It is an evolutionary mismatch of epic proportions. The brain receives this signal and just halts the biological transition tonight.
Wow. So to understand how that signal is communicated to the SCN, we have to look at the neurochemistry. What's the messenger?
The primary excitatory neurotransmitter of the RHT is glutamate. When blue light activates melanopsin, the iPRGCs fire, releasing glutamate directly into the core of the SCN.
So glutamate is the messenger that shouts daytime to the clock.
Precisely.
Which means if you block that messenger, do you deafen the clock?
You do. In experimental settings, if you administer a glutamate receptor antagonist directly to the SCN, you completely block the ability of light to phase shift or entrain the circadian axis. The clock is rendered chemically blind to the environment.
That's incredible. But the body rarely relies on just one pathway. There's usually redundancy. Is glutamate the only input?
No, there's vital redundancy and modulation. There's a secondary visual pathway from the retina that takes a detour through the inner geniculate leaflet, or IGL, before projecting to the SCN via the geniculo-hypothalamic tract.
And what does that one use?
The predominant neurotransmitter in this tract is neuropeptide Y, or NPY, and perhaps most interestingly for psychiatry, there's a prominent projection from the midbrain rapho nuclei that bathes the SCN in serotonin.
Serotonin. Okay, now we're getting into classic psychiatric territory. How does serotonin affect the clock?
It acts as a modulator. It essentially adjusts the volume of the light signal. Current models suggest that serotonin potentiates or strengthens the effect of glutamate during the active phase of the day, making the clock more responsive to light.
Okay, and at night?
Conversely, it inhibits the light signal during the subjective night. It is fine-tuning the pacemaker's sensitivity.
Brilliant. Okay, so we've covered the afferent projections, the inputs. The conductor has seen the blue light baton. Now, how does it direct the orchestra? Let's trace the efferent projections, the outputs.
Well, the majority of the projections from the SCN are local. They terminate within the hypothalamus itself to coordinate autonomic and endocrine functions. But the most thoroughly mapped and conceptually important is the pathway that controls melatonin synthesis.
And this pathway is an anatomical marvel. I tried mapping this out from the textbook, and it is wild. It starts in the brain, goes down into the body, and then has to literally U-turn back up into the head.
It is a highly complex multisynaptic detour.
Walk us through it.
Okay, it begins with SCN neurons, which are primarily GABAergic, meaning they use GABA, an inhibitory neurotransmitter. These neurons project dorsally to the periventricular nucleus, the PVN.
Okay, so from SCN to PVN.
Right. Then from the PVN, long autonomic fibers descend through the brainstem all the way down into the upper segments of the spinal cord.
Way down in the neck area.
Yes. There they synapse with preganglionic sympathetic neurons, which exit the spinal cord and travel to the superior cervical ganglion, a cluster of nerve cells situated in the neck.
Okay.
Finally, postganglionic sympathetic fibers project all the way back up into the skull to innervate the pineal gland.
It's just an incredible amount of evolutionary baggage, isn't it? It takes this massive detour through the sympathetic nervous system, but the key to this whole system is how it acts like a switch.
Exactly. Let's go back to those very first neurons in the SCN. They use GABA. They are inhibitory. This means that when the SCN is highly active, which happens during the bright light of day, it is actively suppressing the rest of the pathway.
It's keeping the brake pedal pressed down.
Spot on.
So during the day, the pineal gland is essentially offline.
Correct. But when darkness falls, the blue light signal stops. The RHT stops releasing glutamate. The SCN quiets down.
And the adrenergic inhibition on the PVN is lifted.
Yes, the brake pedal is released. The signal travels down the spinal cord, up to the neck, and finally to the pineal gland. And at that final synapse, the neurotransmitter released is norepinephrine.
And norepinephrine binding to the pineal gland. And what happens then?
It stimulates beta-1 and alpha-1 adrenergic receptors on the pinealocytes, which triggers a massive surge in the production and release of melatonin into the bloodstream.
And this brings us to a major point of clarification. Because people buy melatonin gummies at the pharmacy and think of them as a natural sleeping pill. They think melatonin equals sleep. But that's a conceptual error, isn't it?
It is a profound oversimplification, and it leads to rampant misuse in clinical and lay populations alike. Melatonin is not a soporific in the way a benzodiazepine is.
Right.
It is the biochemical signal for biologic night. It is a chemical broadcast to every organ, tissue, and cell in the body that it is dark outside. It organizes physiological darkness.
That is a brilliant way to frame it.
In humans who are diurnal, darkness happens to coincide with sleep. But think about nocturnal animals. In mice, the exact same surge of melatonin signals the onset of their highly active waking phase.
Melatonin doesn't mean sleep, it means night. If you're a mouse, night means it's time to run on the wheel. If you're a human, night means it's time to shut down.
Exactly.
Now, if melatonin is the signal for darkness, what's the signal for the dawn?
That would be cortisol. The SEN also projects to the PVN to regulate the hypothalamic pituitary adrenal axis. It targets neurons containing corticotrophin-releasing hormone, CRH.
Which stimulates ACTH from the pituitary.
Correct. Which then causes the adrenal glands to release cortisol. And just as melatonin surges at night, cortisol has a profound circadian rhythm that peaks in the early morning, typically right around 6:0 a.m. to 8:0 a.m.
Right, precisely as melatonin levels are falling back to baseline.
Yes. It's a biological alarm clock. It mobilizes glucose, increases vascular tone, and prepares the organism for the metabolic demands of fighting gravity and hunting for food.
Okay, let's zoom in even further. We've talked about the SCN as a whole organ, but how does an individual microscopic cell inside the SCN actually keep time? I mean, where is the ticking coming from?
We are descending to the level of the molecular clockwork now, and the sheer elegance of this genetic machinery is astonishing.
I'm ready.
The mammalian circadian clock within each of these cells is driven by interacting positive and negative transcriptional and translational feedback loops.
Okay, let's break that down without getting lost in the alphabet soup of genetics. Imagine the cell as a factory. How does the factory run on a 24-hour shift?
Let's start with the positive limb, the engine of the factory. Inside the cell nucleus, you have a protein complex made of two proteins bound together, CLOCK and BMAL1.
CLOCK and BMAL1.
Yes. Think of this CLOCK-BMAL1 complex as the master switch. It binds to the DNA and activates the transcription of several specific worker genes. The most important of these are the period genes, or PER, and the cryptochrome genes, or CRY.
Okay, so the master switch is thrown, the DNA is read, and the factory starts churning out PER and CRY messenger RNA. And that RNA goes out into the cell to build PER and CRY proteins.
Exactly. As the morning progresses into the afternoon, the levels of PER and CRY proteins steadily build up in the cytoplasm outside the nucleus. They are accumulating on the factory floor.
Okay.
Eventually, usually by the evening, they reach a critical mass, they bind together into large complexes, and then they do something remarkable. They translocate back into the nucleus.
Your workers head back to the boardroom.
I love that. Yes. And when they get there, they physically interact with the CLOCK-BMAL1 master switch and rip it off the DNA. They repress their own transcription.
This is the negative feedback limb.
Yes. The products of the factory shut down the factory itself.
So no more PER and CRY are made. The machinery halts.
Meanwhile, the CLOCK-BMAL1 complex also activated a gene called RevERB alpha. This protein enters the nucleus and represses the transcription of the BMAL1 gene itself.
So the whole thing just goes quiet.
The entire system is in a state of profound inhibition throughout the biological night. However, these PER and CRY proteins are not stable forever. Over the course of the night, they gradually degrade. As the sun begins to rise, the last of the repressor proteins are cleared away. The inhibition is lifted, BMAL1 is produced again, binds with CLOCK, and the 24-hour cycle begins anew.
It is a chemical hourglass. It takes about 12 hours for the proteins to build up and shut the system down, and about 12 hours for them to degrade and let it start over.
Beautifully said.
But it can't just be a passive build-up and breakdown, right? There has to be a way to speed it up or slow it down to fine-tune that 24.19-hour rhythm to exactly 24 hours.
You are absolutely right. The transit time, how quickly these proteins move between the cytoplasm and the nucleus, and how quickly they degrade, is exquisitely regulated by post-translational modifications.
Quality control managers.
Exactly. Specific enzymes act as quality control managers. For instance, casein kinase 1 epsilon and casein kinase 1 delta attach phosphate groups to the PER proteins.
Okay, so they phosphorylate them.
Yes, this phosphorylation is a molecular tag. It dictates whether the PER protein is allowed to enter the nucleus or if it is immediately targeted for destruction by the proteasome. If you alter those kinases, you drastically alter the speed of the clock.
Which means if any of these genes mutate, if the PER gene is malformed, or if the casein kinase enzyme is overactive, the hourglass empties too fast or too slow.
And the clinical implications of that are massive.
Well, before we get to the pathology, let's look at how this perfect genetic clock translates to the thing we spend a third of our lives doing: sleep.
Sleep is the most prominent output of the circadian system, but it is frequently misunderstood clinically. Many view sleep as a simple neurological switch, a flip from on to off.
Just powering down.
Right. But in reality, sleep is the integrated, dynamic product of two distinct, powerful processes. We call this the two-process model of sleep regulation.
The two heavyweights in the ring: Process S and Process C. Let's look at Process S first.
Process S represents the sleep homeostat. Think of this as your biological sleep debt. The moment you wake up in the morning, this drive for sleep begins to climb, and it accumulates steadily for every hour you remain awake.
And biochemically, what's driving that?
While the exact biochemical substrates are still being mapped, adenosine is considered a primary neuromodulator of this drive.
Okay, let's follow the adenosine, because as the brain burns energy throughout the day using ATP, adenosine is left over as a metabolic byproduct. It just starts building up in the brain.
Specifically, it accumulates in areas like the basal forebrain and the ventrolateral preoptic nucleus, or the VLPO.
The VLP.
Yes. The VLPO is essentially the brain's sleep switch. As adenosine binds to its receptors here, it exerts an inhibitory effect on the wake-promoting regions of the brain. It creates an increasing pressure, an overwhelming urge to sleep.
And this is exactly how the most widely consumed psychoactive drug in the world works, isn't it? Caffeine.
Yes, caffeine is an adenosine receptor antagonist.
It slips into those receptors in the VLPO but doesn't activate them. It just blocks the adenosine from docking. So the brain doesn't feel the weight of the sleep debt, even though the adenosine is still piling up behind the dam.
A perfect description of the pharmacological illusion of caffeine. So that is process S, the homeostatic pressure pushing you down into sleep.
Right.
But if process S were the only mechanism, you would be progressively sleepier every hour of the day until you collapsed. Yet most people feel relatively alert at, say, 6:00 p.m., despite having been awake for 10 hours. Why? Because of process C.
The circadian counterpunch.
Exactly. Process C is the circadian component, driven by our master conductor, the SCN. Rather than promoting sleep, process C actively promotes wakefulness. And crucially, the strength of this wake-promoting signal steadily increases throughout the subjective day.
Okay, I visualize this as a stretched rubber band.
Oh, how so?
Process S is you stretching the rubber band further and further all day long. The tension is massive. But process C is a hand holding the other end of the rubber band, pulling back in the opposite direction.
Yes.
Process C actually reaches its peak intensity in the late evening, the wake maintenance zone, fighting off the crushing sleep debt you've accumulated. But then as darkness falls and the SCN registers the drop in light.
The SCN quiets down. Process C lets go of the rubber band. The circadian wake promotion rapidly declines, and the massive accumulated weight of process S plunges you deep into sleep.
And during the night, as you sleep, process S is cleared, the adenosine is metabolized.
Concurrently, process C remains at its trough.
And right in the middle of that trough, our core body temperature hits its absolute lowest point, the nadir.
Yes. The temperature nadir typically occurs between 5:00 a.m. and 6:00 a.m. in a standard chronotype. This timing is mathematically critical for sleep architecture.
Mathematical.
Yes. Experimental protocols have demonstrated that an uninterrupted, consolidated 8-hour sleep bout is most successful when it is initiated approximately 6 hours before this temperature nadir.
So if my nadir is at 5 a.m., I should theoretically be falling asleep right around 11:00 p.m. That aligns my homeostatic clearance perfectly with the trough of my circadian arousal.
It creates the optimal temporal window. But we must recognize that this window changes across the lifespan.
Right. You often hear the colloquialism that older individuals simply need less sleep, or that waking up 5 times a night is just a normal part of getting older.
We really need to dissect that carefully.
Let's dissect it, because looking at the textbook data, sleep fragmentation in the elderly seems much more pathological than just normal aging.
There is a genuine age-dependent decline in objective sleep need from infancy through early adulthood, and it plateaus in midlife. However, the profound sleep fragmentation and loss of deep, slow-wave sleep seen in many geriatric patients is not entirely benign.
So what's causing it?
Research indicates it is closely tied to the physical degeneration of those VLPO neurons we just discussed, the very cells responsible for integrating process S and sustaining sleep. As those neurons die off, the homeostatic drive loses its grip.
It's an anatomical breakdown.
It is. Furthermore, this fragmentation is massively exacerbated by polypharmacy, nocturia, pain, and psychosocial isolation. Dismissing severe insomnia in an 80-year-old as just aging is a failure of clinical reasoning.
It's an anatomical breakdown, not just a lifestyle shift. And that transition from physiology to pathology is exactly where we need to take this conversation next.
Into the clinical manifestations.
Exactly. We've mapped the Burkett Orchestra. Now, what happens when sections of the orchestra go rogue? What happens when the rhythm breaks? And the most glaring example of a broken rhythm is seasonal affective disorder.
Seasonal affective disorder, or SAD, represents the intersection of environmental light and mood regulation. It is important to note that in the DSM-5, SAD is not a standalone diagnostic entity.
Right, it's a specifier.
Yes. Major depressive disorder, bipolar first or bipolar thec, with seasonal pattern. It is remarkably common, with a prevalence ranging from 4% to 9%, depending on latitude. And it carries a strong genetic vulnerability.
How strong?
Twin studies suggest the heritability of seasonality is between 29% and 43%.
Wow. That is a staggering genetic load for a condition ostensibly triggered by the changing of the seasons.
Triggered specifically by the photoperiod, the duration of daylight. The fundamental mechanism driving winter SAD involves what chronobiologists call the long night profile.
The long night profile.
Yes, in the winter, as the days shorten and the nights lengthen, the biological duration of elevated melatonin secretion from the pineal gland expands. The brain is chemically registering a longer period of darkness.
Okay, I want to play devil's advocate here based on the clinical treatments. The standard narrative is the phase delay hypothesis. The sun comes up later in the winter, so the circadian clock drifts late. It's misaligned.
That's the classic view.
And if that is the sole cause, then administering bright light therapy early in the morning should act as an artificial sunrise, truncate that long melatonin profile, and instantly cure the depression. And to be fair, morning light therapy is highly effective.
It is.
But the textbook points out a glaring paradox for many patients. Bright light therapy in the evening also works. If they are already phase delayed, shouldn't evening light delay them even further and make them worse? Why does evening light work?
You have zeroed in on a crucial theoretical debate. The efficacy of evening light therapy severely challenges the pure phase delay hypothesis. If misaligned timing were the only issue, evening light should indeed exacerbate the pathology.
So what's the alternative?
This leads us to the competing theory, the photic insensitivity hypothesis.
Explain photic insensitivity. What does that mean for the patient?
It posits that individuals vulnerable to SAD have a fundamental biological hyposensitivity to light. During the summer, ambient light levels are so incredibly high, often 100,000 lux outdoors, that even with a dampened sensitivity, their system receives enough photic input to maintain euthymia.
Oh, okay. But in the winter?
In the winter, when individuals spend most of their time indoors under artificial lighting of maybe 300 lux, the input drops below their specific biological threshold. The circadian system is effectively starved of light.
Ah, so it's not that the clock is necessarily set to the wrong time, it's that the clock is fading away because the battery is dying.
Precisely. And in this model, bright light therapy, whether administered in the morning, midday, or evening, is not just shifting the clock. By sitting a patient in front of a 10,000 lux light box for 45 minutes, you are brute forcing the system.
Brute force in the biology.
Yes. You are delivering a massive payload of photic energy that crosses their elevated threshold and restores the amplitude of the circadian system, regardless of the precise phase shift it induces.
It makes sense why 10,000 lux is the gold standard. You need a sledgehammer, not a scalpel.
Exactly. And while we're talking about SAD, we shouldn't overlook that targeted cognitive behavioral therapy, CBT, specifically adapted for SAD, focusing on behavioral activation and sleep hygiene, shows remarkable efficacy, particularly in preventing relapse in subsequent winters.
But what about depression that doesn't care what season it is? Non-seasonal unipolar depression. How is the clock involved there?
The chronobiological footprint is everywhere in melancholic depression. We consistently observe a dampened amplitude of diurnal rhythms. The physiological peaks and troughs are flattened.
What does that look like physiologically?
The core body temperature doesn't rise as high during the day or fall as low at night. The nighttime melatonin surge is blunted. Furthermore, the architecture of sleep itself is distorted.
The textbook highlights a classic polysomnographic finding in depression, shortened REM latency. A healthy person might take 90 minutes to enter their first cycle of REM, or dream sleep. But a severely depressed patient might hit REM in 40 minutes, and the density of their eye movements during REM is dramatically increased. The timing of sleep stages is completely compressed.
And this chronobiological dysregulation leads us to one of the most counterintuitive, paradoxical interventions in all of psychiatry wake therapy.
I am continually blown away by this concept. You have a patient with severe, crushing depression. They are exhausted, anhedonic, perhaps actively suicidal. And the intervention is to completely deprive them of sleep. You keep them awake all night, or at least for the second half of the night. It sounds like a medieval torture method, but the clinical data is undeniable. In about 60% of these patients, total sleep deprivation produces a rapid, profound antidepressant response within 24 hours. How is that biochemically possible?
To unravel the paradox, we return to the two-process model. Current theory suggests that in certain depressive subtypes, the homeostatic drive process S may be functioning abnormally, perhaps failing to accumulate properly or interacting erratically with the circadian pacemaker.
Okay.
By forcing the patient to remain awake, we are forcefully driving up that homeostatic pressure to a massive degree. We are artificially loading the system with adenosine and other somnogens, which somehow overrides the depressive neural circuitry and temporarily resets the neurobiological state.
But the textbook is very explicit. There is a monumental catch to this miracle cure.
A devastating catch. The antidepressant effect of wake therapy is almost universally transient. The moment the patient is allowed to have a recovery sleep, even a brief nap, the profound depressive symptoms rapidly return upon waking. Therefore, wake therapy cannot be utilized as a standalone definitive treatment. It is a tool for rapid, acute relief. But to sustain that clinical response, it must be immediately combined with phase-advancing the sleep cycle, implementing bright light therapy, or initiating rapid-acting pharmacological antidepressants.
It's like jump-starting a dead car battery. It gets the engine running for a minute, but if the alternator is broken, the moment you turn it off, it's dead again. You have to fix the alternator while the engine is running.
A very apt metaphor.
And while we are on the topic of massive mood shifts, we have to talk about bipolar disorder. If unipolar depression is a flattened rhythm, bipolar disorder is a rhythm swinging violently out of control.
Bipolar disorder is arguably the psychiatric condition most intimately linked to circadian dysregulation. The sleep-wake disruptions are not merely secondary symptoms. They are core features of the pathophysiology.
To understand this connection at a molecular level, the text points to a landmark animal model, the clock delta 19 mouse. Let's unpack this.
Okay. Researchers took the clock gene, one half of that master switch in the molecular factory we talked about earlier, and they intentionally mutated it. They deleted a specific section, creating the clock delta 19 mutation.
Now, I understand how breaking a clock gene messes up a mouse's sleep schedule, but how on earth does a broken clock gene result in a mouse exhibiting manic-like behaviors? Hyperactivity, extreme risk-taking, decreased need for sleep. How do you get from a broken watch to clinical mania?
It is a remarkable leap, and the answer lies in the neuroanatomical integration of the circadian system with the brain's reward circuitry. The SCN doesn't just dictate sleep, it modulates vast networks.
Right.
In these clock delta 19 mutant mice, researchers discovered a profound abnormality in the ventral tegmental area, or the VTA.
Ah, the VTA is the heart of the brain's dopamine system. It's the engine of reward, motivation, and salience.
Exactly. And the researchers found that the clock gene acts as a negative regulator of dopaminergic activity in the VTA. When the clock gene is mutated and dysfunctional, that regulation is lost. Dopamine synthesis and firing rates in the VTA surge dramatically.
So a broken circadian gear leads directly to an overflowing dopamine system.
Yes. And the definitive proof of this mechanism is elegant when researchers use viral vectors to deliver functional, unmutated CLOCK protein exclusively into the VTA of these mutant mice, leaving the rest of the brain's clock broken. It completely rescued the manic-like behavioral phenotype.
The hyperactivity ceased just from fixing the clock in the VTA?
Yes, this explicitly links the circadian modulation of dopaminergic circuitry to the core features of mania.
That completely reframes how we look at a manic patient. It's not just a mood episode. It's a profound neurochemical dysrhythmia. And in human bipolar patients, we see intense state-dependent shifts in their circadian phase.
We do.
When they are manic, their rhythms are severely phase-advanced. They are waking up at 3 a.m. full of energy. When they crash into bipolar depression, their rhythms become severely phase-delayed. They can't drag themselves out of bed until the afternoon.
Because the biological clock is inherently fragile in these individuals, behavioral interventions that externally stabilize the rhythm are profoundly therapeutic. This is the rationale behind interpersonal and social rhythm therapy, or IPSRT.
What does that involve?
It is a structured psychotherapy that focuses on rigidly routinizing social zeitgebers. Patients are trained to eat, wake, exercise, and socialize at the exact same times every single day.
It's providing an external scaffold for a broken internal clock, and the clinical outcomes are incredible. Research shows that IPSRT can be as effective as pharmacological mood stabilizers in preventing relapse, particularly for bipolar II disorder.
This is highly effective.
But there's a vital warning label regarding chronotherapy in bipolar patients, isn't there? We talked about brute-forcing SAD with light therapy. Can we use a 10,000 lux light box to treat bipolar depression?
You must proceed with extreme clinical caution. Because bright light therapy forcefully shifts the circadian pacemaker, administering it to an individual with bipolar disorder carries a significant risk of abruptly unmasking manic tendencies and precipitating a severe manic or hypomanic episode.
Oh, wow.
The circadian system is highly reactive. If light therapy is indicated for bipolar depression, it is generally safer to administer it at midday rather than morning, and ideally, only when the patient has concurrent pharmacological mood stabilization on board. You cannot forcefully yank the circadian system of a bipolar patient without protection.
That is a critical takeaway for any clinician prescribing light therapy. Okay, let's pivot to the final major clinical area, neurocognitive disorders, specifically dementia and aging. We know that as the brain physically degenerates, cognition fails. But the temporal architecture fails too.
It fails profoundly and in pathology-specific ways. The characteristic rest activity patterns shift depending on the specific neurodegenerative disease.
Give us some examples.
Well, in Alzheimer's disease, we commonly observe a pronounced phase delay. Patients remain awake late into the night. Conversely, in frontotemporal dementia, we frequently see a phase advance. And in diseases affecting the basal ganglia and brainstem, such as Parkinson's disease or progressive supranuclear palsy, we can witness a complete sleep-wake reversal.
Where they are awake all night and asleep all day.
Exactly. Insomniac entirely through the night and hypersomnolent throughout the day.
Now, the textbook offers a striking insight here that completely flipped my perspective. We usually think of a dementia patient wandering the halls at 2 a.m. as a sad symptom of their declining memory. But the evidence suggests that circadian disruption actually predates the clinical diagnosis of Alzheimer's disease.
Yes.
It's not just a late-stage symptom, it predicts the onset of the disease.
It is a paradigm-shifting revelation in neurology. The circadian dysregulation may not merely be a downstream consequence of neuronal death. It may play a central, mechanistically causative role in the pathogenesis.
How so?
We know that the glymphatic system, the brain's waste clearance mechanism that flushes out toxic beta-amyloid and tau proteins, is highly active during deep sleep. If the circadian rhythm breaks down early in the disease process, sleep is fragmented, clearance fails, and the toxic proteins accumulate faster, accelerating the neurodegeneration.
The broken clock is driving the disease. And clinically, in a residential care setting, this temporal breakdown manifests as a phenomenon we call sundowning.
Sundowning is the exacerbation of confusion, agitation, wandering, and aggression that predictably occurs in the late afternoon and early evening in patients with dementia. This is deeply correlated with the disruption of circadian-regulated cholinergic functioning in the basal forebrain.
So how do we intervene? We can't cure the Alzheimer's, but can we fix the clock?
We can try to reinforce it by implementing robust daytime bright light therapy, increasing ambient illumination in the facility. During the day, we strengthen the photic Zeitgeber signal to whatever functional SCN tissue remains.
Does it work?
Clinical trials have demonstrated that this can consolidate nighttime sleep, reduce nocturnal wandering, and remarkably, slightly improve objective daytime cognitive scores. We are using light to restore a modicum of temporal order to a failing brain.
Which is a beautiful integration of biology and environment. And this integration brings us seamlessly to our final major topic, section 5 of the chapter, chronopharmacology, the intersection of time and medication.
This is crucial.
We need to look at this bidirectionally. How do drugs alter the clock, and how does the clock alter the drugs? Let's start with the drugs affecting the clock.
Many of the psychiatric medications we prescribe exert profound and often unappreciated effects on the circadian pacemaker. We noted earlier that patients with melancholic depression exhibit a flattened circadian amplitude. When we prescribe antidepressants, such as tricyclic antidepressants or SSRIs, part of their therapeutic efficacy appears to be chronobiological. They actively reduce the abnormally elevated nocturnal body temperature and enhance the amplitude of the daily rhythms. They act as pharmacological timekeepers, restoring the robust peaks and troughs.
So antidepressants fix the clock, but other drugs can smash the clock. The text specifically highlights short-acting benzodiazepines like triazolam.
Triazolam is fascinating chronobiologically. If administered to an animal model during the middle of their subjective active phase, it can induce significant circadian phase advances. It chemically fast-forwards the clock.
Oh.
At a molecular level, triazolam has been shown to rapidly reduce the light-induced transcription of the PER1 and PER2 clock genes inside the SCN. It is reaching directly into the molecular factory and disrupting the gears.
What about recreational drugs? We talked about serotonin modulating the light signal. What does MDMA ecstasy do to the system?
MDMA acts as a profound serotonin neurotoxin. Repeated exposure physically damages and destroys the delicate serotonergic axonal terminals projecting from the roughened nuclei into the SCN.
Oh, wow.
By severing this connection, MDMA abolishes the clock's ability to modulate it by serotonin. The clinical consequence is that the individual loses the ability to rapidly re-entrain to new light-dark cycles, leading to persistent, enduring circadian dysrhythmia long after the drug has left their system.
A terrifying consequence of destroying the volume knob on the light signal. Okay, so that's how drugs alter the clock. Now let's flip the paradigm. How does the clock alter the drugs? This brings us back to the hook we opened the episode with, chronotherapeutics.
Chronotherapeutics is the principle that the efficacy, pharmacokinetics, and toxicity of medications fluctuate significantly based on the time of day they're administered. This is particularly vital in oncology.
Right, the cancer drugs.
In animal models, the lethal toxicity of fixed doses of various anti-cancer agents can vary by a factor of 2 to 10-fold simply based on the circadian phase of administration.
A tenfold difference between a cure and a lethal overdose, entirely dependent on the hour. Walk me through the physiology of why that happens. Why does a drug care what time it is?
Because every physiological parameter required to absorb, distribute, metabolize, and excrete a drug is governed by the circadian clock. Gastric pH fluctuates, fundamentally altering the absorption of orally administered weak acids or bases. Gastrointestinal motility is rhythmic, changing transit times.
What about the kidneys?
The glomerular filtration rate in the kidneys, which handles the elimination of many drugs, operates on a profound circadian rhythm, peaking during the active phase. Even the physical viscosity of cellular membranes changes throughout the day, altering how lithophilic drugs distribute into target tissues.
And liver enzymes, the cytochrome P450 system must have a rhythm.
Absolutely. The expression and activity of hepatic metabolic enzymes rise and fall. If you administer a highly toxic, narrow therapeutic index drug at the exact moment when the liver's metabolic enzymes are at their circadian trough, the drug accumulates rapidly and you induce severe, potentially fatal toxicity.
It's an undeniable mandate that we have to prescribe the minute as carefully as we prescribe the milligram. Finally, let's look at how we use chronotherapeutics to treat primary circadian rhythm sleep-wake disorders.
We must carefully distinguish between the two primary phase disorders. On one end, we have advanced sleep phase syndrome, or ASPS. This is the extreme lark. They cannot stay awake past 7:30 p.m., and they wake up irrepressibly at 3:30 or 4 a.m.
Is that genetic?
A subset of this is familial advanced sleep phase syndrome, FASPS, which is directly linked to an autosomal dominant genetic mutation in the PR2 gene. The mutated PR2 protein alters the kinetics of the molecular clock, drastically shortening the period length. The clock spins too fast.
And on the opposite end, we have delayed sleep phase syndrome, DSPS, the extreme owl unable to fall asleep before 3 a.m., sleeping until noon. This is frequently associated with a polymorphism in the PER3 gene. Their clock runs too slow. Now, when a patient with a delayed phase comes into the clinic, the first thing they usually say is, I took a massive dose of melatonin at bedtime and it didn't work. And the textbook puts a massive flashing warning sign right here about how we use exogenous melatonin.
And it is a vital clinical lesson. Melatonin is wildly over-the-counter in many countries, often sold in massive supraphysiologic doses of 5 or 10 milligrams. The intuitive logic is to take it precisely at bedtime, treating it like a standard hypnotic sleeping pill.
But we established earlier, melatonin isn't a sleeping pill. It's the signal for biological night. And in a natural, healthy system, the body's own production of melatonin, what we call the dim light melatonin onset, or DLMO, starts rising 2 to 4 hours before habitual sleep onset.
Exactly. The brain needs a gradual twilight, not a sudden midnight. If you flood the system with a massive 10 milligram spike of exogenous melatonin precisely at the moment of sleep onset, you run a high risk of sending a confusing, mistimed phase-shifting signal to the SCN.
With what result?
Depending on their exact phase response curve, you can inadvertently induce a phase delay relative to the environment. You are effectively giving the patient artificial jet lag every single night, exacerbating their underlying circadian dysregulation rather than fixing it.
So if you're using melatonin as a chronobiotic, a tool to shift the clock rather than just forcing drowsiness, the dosing has to be tiny, like 0.5 milligrams, and the timing has to be exquisitely precise, usually several hours before the desired bedtime.
Precise timing is the essence of chronopharmacology. Okay, we have traversed from molecular genetics to clinical prescribing. It is time to synthesize our core mental models, and we're going to do this by walking through 5 rapid-fire clinical scenarios. I want you, the listener, to use the clinical reasoning we've just built to puzzle through these cases with us. Pause if you need to think.
Let's do it. Case number 1. Imagine a 45-year-old patient who works entirely night shifts. They are suffering from severe shift work disorder, constantly fighting a misaligned circadian phase. A pharmaceutical rep visits your clinic pitching a novel, targeted medication designed to selectively block the primary photic input to the SCN. They claim it will blind the clock to unwanted morning light so the night worker can sleep. Based on the neuroanatomy we discussed, what specific neurotransmitter pathway originating in the retinohypothalamic tract is this drug most likely antagonizing?
So if the goal is to sever the visual light signal originating from the melanopsin-containing ganglion cells, the drug must target glutamate. Glutamate is the primary excitatory neurotransmitter released by the RHT into the core of the SCN. Block the glutamate receptors in the SCN, and the circadian clock no longer registers environmental light.
Excellent. Case number 2. An 82-year-old patient is brought in by their family. The family reports the patient sleeps in scattered 1-hour bursts throughout the day and night, accumulating maybe 4 total hours. It is severe consequential sleep fragmentation. While assessing them, you recognize this goes far beyond the normal age-related reduction in circadian amplitude. You suspect a structural breakdown in the homeostatic sleep drive itself, which underlying neuroanatomical degeneration most directly accounts for this inability to maintain sleep pressure.
We're looking at the failure process S here. The structural origin for the homeostatic sleep switch is the ventrolateral preoptic nucleus, or VLPO. Physical loss and degeneration of the inhibitory neurons in the VLPO directly strips the brain of its ability to consolidate sleep and respond to adenosine accumulation. It is a profound structural deficit, not merely a chicken timing.
Spot on. Case number 3. A patient with a 10-year history of severe treatment-resistant winter seasonal affective disorder is finally prescribed a rigorous daily regimen of 10,000 lux morning bright light therapy. To explain the mechanism to the patient, you need to describe exactly what the light is doing to their neuroendocrine system. What specific physiological parameter of their endogenous melatonin secretion is this morning light attempting to target and correct?
The clinical target is the long night profile. In winter SAD, the duration of melatonin elevation extends abnormally into the morning hours. High-intensity morning light therapy acts as an artificial early dawn, striking the RHT and acutely truncating that extended tail of melatonin secretion, rapidly signaling to the body the biological day has begun.
Brilliant. Case number 4. A 28-year-old patient presents complaining of insomnia. However, upon reviewing their sleep diary, they don't have trouble sleeping. They have trouble staying awake. They are completely exhausted and fall asleep irrepressibly by 7:30 p.m., waking naturally and fully alert at 4 a.m. They report their father and grandmother had the exact same sleep pattern. You order genetic testing. A mutation in which core molecular clock gene is most classically associated with this presentation of familial advanced sleep phase syndrome.
The familial autosomal dominant presentation of an extreme advanced phase is classically associated with the mutation in the PER2 gene. This structural alteration to the period 2 protein accelerates the clearance of the negative feedback loop in the molecular clock, forcing the entire endogenous cycle to run significantly faster than 24 hours.
And our final case, number 5. A patient with a known diagnosis of bipolar I disorder is admitted experiencing a severe manic episode characterized by extreme hyperactivity, grandiosity, and a profoundly decreased need for sleep. You are considering the chronobiological underpinnings of this presentation. Based on insights drawn from the clock delta 19 mouse model, dysregulation of which neurotransmitter in which specific brain region is most strongly implicated by core circadian gene mutations driving manic behavior.
The clock delta 19 model explicitly links circadian dysfunction to the reward circuitry. The mutation drives a massive surge in the synthesis and transmission of dopamine, specifically within the ventral tegmental area, or VTA. Restoring normal clock function exclusively in the VTA rescues the manic phenotype, proving that circadian regulation of dopaminergic tone is central to the pathophysiology of bipolar mania.
And that ties the genetics, the anatomy, the pharmacology, and the clinical presentation together perfectly. We have reached the end of our synthesis.
I want to leave you with one final provocative thought. We discussed how a drug's toxicity and efficacy can vary tenfold simply based on the hour it is administered. Consider the vast history of clinical trials in psychiatry. How many potentially revolutionary life-saving molecules were abandoned in phase 2 trials because they showed lack of efficacy or unacceptable side effects?
Right.
How many of those failed psychiatric medications didn't actually have the wrong molecular structure, but were simply administered at the wrong biological time? We have spent a century treating the human mind as a static target, when in reality we are treating a moving target as if it were standing still.
That concept completely transforms how you look at a prescription pad and how you look at the patient sitting across from you. Thank you for joining us on this exploration. We encourage you to return to the textbook, read the chapter with this new framework, and keep looking at the clock. Here are the details of this episode.
Chronobiology, circadian rhythm, and psychiatry. Byline: the rhythm of the mind, how time itself structures mood, cognition, and behavior.